basis do we choose which cohorts we have to use?
Purely numbers. So if you've got 5 data sets, which have got over 200 individuals, use them all and see what is common between those 5 data sets. Because the more you look at these, the more data you look at, the more and cross-validations you do, the more robust the things you will find are. And that's that cross-validation that's really important. If you consider sort of 6,000 individuals across 5 cohorts compared to 100 individuals over 2 cohorts, that result is going to be much stronger for the bigger number. So, yeah. Okay. Any other questions? Is there anything anybody wants me to clarify, go over again? Can you see? We modelled the p53 pathway. We worked out the drivers of the p53 pathway. And then we looked at how druggable those drivers were, and if there were other molecules which we might be able to utilize to target them in the pharmacopoeia, the body of knowledge around pharmaceutical entities. So the question here ultimately is, can we develop an in silico pipeline for drug discovery that leverage the power of omics data, big data, and machine learning? In other words, can we digitize drug discovery? So again, this relies heavily on this distribution curve, which helps us with false discovery issues, and we can use this method to enrich by concordance and stability. Except now, rather than using it to answer a question around phenotype, so rather than say using it to predict healthy versus lung cancer, we're now using it to predict a gene in a pathway. So each one of those, each one of these green boxes now becomes an output for a neural network. So I'll show how we do that in a little bit more detail. Okay. So rather than showing the whole p53, what's going on here? Never mind, we'll see. Rather than showing the whole p53 pathway, which would look really complicated, I'm just creating a simplified network to show how we do this. So let's say we have a hypothetical pathway where molecule X is known to link to Y, which is linked to Z. Molecule X links to B, which links to C. Molecule X links to molecule A. So that's a simple pathway. Each one of these blue dots is a gene, and they're thought to link together in a molecular pathway. So what we can do then is we can then start to mine the interactors for each one of these members of the pathway using this approach of looking at distribution curves. And we can mine the whole transcriptome to see what things strongly predict that target in that condition. So we can start doing this mining for a breast cancer data set. So we find the interactors of molecule A or X. We then do the same thing. So it's going a bit slow at the minute. I have a whirly wheel on the screen. I don't know why I'm going to go slow. So we then do the same thing for the next molecule. The next molecule. So effectively, sorry, it's going very slowly. So effectively, we map the interactome for every node in the whole pathway. So you're gaining evidence about how things are interacting using the known molecules of the pathway as a seed to build a network up. What we then do is enrich using this distribution curve. So we now have a set of stable interactors for each gene in the pathway. So what we then do, sorry, this is kind of animating itself because it's going so slowly, is we then look for commonalities between those stable interactomes. So a red dot is something that's found in the interactome of this one and this one, the stable interactome. So effectively, with this new red dot here is a new gene that is an intermediary in the pathway based upon, sorry. Computer's going wild. I don't know what's happening here. Right, I'm just going to use this way to present. It's less slow. So, as I say, we've got a stable interactome for each member of the pathway. We look for commonalities, and effectively we're gap filling the p53 pathway in breast cancer, so it's highly specific, and but by searching across the whole transcriptome. So in this way, we are effectively gaining new knowledge about that pathway and what might be involved in it with it. If you look at other methods, and if you remember I talked to you about Gene Mania, Metacore, Ingenuity, those pathways are generic. They kind of look at all of biology. You might be able to get a bit of information around certain conditions, but they're generic. They kind of give the vanilla flavor, general biology, whereas this builds the network specifically from omics data, specifically for a subset of cases. So that's useful because it means we can quantify and add to the network. So what we did here is we used this Metabrick data that I mentioned before, and we looked at a clinically annotated primary invasive breast cancer set, specifically focusing on the triple negative. So you may have heard of a triple negative breast cancer. It's ER, PR, HER2 negative. Some histologically call it a basal phenotype. So that was the data set, and we looked at TCGA. And we specifically selected triple negative cases. So when we mined the p53 pathway, everything, let me just get this the right way around, everything in green here, everything in green here is a new feature of that pathway that's come up based upon evidence from the mining of the transcriptomic data. So these are new things. The pathway is more complicated than we realized, And using this method, we can add features that are specific to cancer into that system. So what we then did is used inference modeling. And I've already talked about this. But the same process is applied. And what we get to, like we did before, but now slightly differently, is an augmented p53 pathway. So we can see this is a tissue necrotizing factor, this is EGFR. We're getting strong hubs again, that like what we found before. So these are things that are highly influenced in the p53 system in breast cancer. But as I said before, we can do a driver analysis and work out how much influence and how influenced each feature is. So this identifies the most influential molecules. We've done previous studies where we've shown that the most influential molecules, if we change them, manipulate them, we can change phenotype. I'll show some examples from plant sciences in a bit where we created transgenic plants by switching certain transcription factors on and off. So that gave us an indication that these drivers and hubs are interesting things and potentially are interesting for drug targets, as drug targets. So the Lancet paper, we showed those methods there, and we found actually in the Lancet study, when we created a knockout, I'll show this in a second, of the SPAG5, we actually slowed proliferation right down and we changed the centromere structure of the cells. So again, phenotypic changes that resulted from that target. So, as I said before, driver analysis takes the sum to each gene and tells us the sum of genes leading to a gene and tells us how influenced the gene is, and then takes the sum of the genes links leading from the gene to tell us how influential it is. And what's that? This is the one, yeah. The order of this one, I think the top most influential features, there's a disconnect between the name and the gene, but this is the correct value. So STEAP3, CD82, TP73, those were the most influential. These are novel. So these top three are known in the pathway. Just ignore this bit. These two are novel. And we're finding highly influential novel features coming up. If we look at the most influenced features, the names are correct here. So what's interesting here, if we look at the most influenced features, we have our number 2 feature, which is known, and that's a TP53 apoptosis inducing protein. So we've gone through the whole transcriptome, looked at everything, looked at the network inference, and we find features that make biological sense, being highly influenced and highly influential in the system we're exploring. So that gives us some confidence that we're starting to unravel some of the biology here with this method. Now, we can do the connectivity and stability. I've already talked to you about that. Once we have that, we can start to interrogate the databases that are out there for things that might be interesting in terms of their druggability. What we can also do, though, and this is important, is because we make the model and do the network inference on a specific type of data, so triple negative breast cancer cases, we can also repeat that process for healthy breast, for healthy lung, for healthy kidney, liver, skin, blood. And we can basically build differential models of the disease state, from the disease state, of the pathway in different tissues. And that's moving towards something called a digital twin, where we can build effectively a model of different parts of an individual and see how the pathway works and how it changes between those individuals, or not between the individuals, between the parts of an individual. And that's important for drug discovery because firstly, we don't want to drug biology that's essential for healthy life. If we start knocking out essential systems, then people have serious side effects and die from the drug. But if we knock out things in the cancer that are not present in the healthy state, then we can start to find useful things that are differential from healthy. Finding, if you like, the molecular causality of the disease. So causality is what we're looking for here. What's the molecule that causes this problem? Or what are the molecules that cause this problem? So once we have a target, we can drop the target into DGIDB, the Drug Genome Interaction Database. That uses a combination of curation, so curated data sets, and curated databases, and data have been mined from DrugBank, from PharmGKB, from Chembol, and from the Drug Target Commons. So these are all sources of drugging information for molecules. So genes are here categorized as potentially druggable, based upon their membership in pathways, functions, families, ontologies, the Human Protein Atlas, and the indicative druggable genome. So this is found at DGIGB. So rather than showing you some results, I'm going to use this as an example. So let's pick a strong driver, GAD45B. So if I get I go to search DGIDB. This is the Drug Genome Interaction Database. So let's pick some things from this list. So let's say GAD45B, that's a starting one. And then let's pick another one, TP53 AIP1. And I'll pick a third. I could put up to 50 in here if I wanted to, or more than 50. Let's do TP7, integrin beta 1, ITGB1. I'll pick that one. ITGB1. Yep. And I'll search. So it's picked up 3 genes, it's found them. There's 4 potential interactions that are inhibitors. there were 3 potential interactions that are antibodies. And this is our table of results. So what I'm interested here is in interaction score. So I'll sort by interaction score, largest to smallest. So first one, integrin beta 1. So this is volociximab. Sorry, you have to get your tongue around databases as drug names sometimes. So volociximab is an anti-neoplastic agent. So that's quite good because neoplasms are cancer. So this is good for treating cancers. Integrin beta 1 is a target. It's not been approved for that target, but it has a pretty strong interaction score. So I could take, let's say I want to find triple negative breast cancer, I could suggest, given the role of this molecule being strongly influenced in breast cancer, let's try a trial where we look at volociximab and its treatment for breast cancer. I can see more about volociximab. So let's see what I can get from that. So volociximab, well, mab at the end of a drug means monoclonal antibody. So volociximab is a monoclonal antibody targeting this biology. It's got a Kemble identity. It's been used up to phase 3, but there's not any FDA data linked. There were other interactions with integrin alpha 5 as well. So that's a potential target. Let's see what else we've got. So we've got, these are unique matches. GAD doesn't seem to be any good. It's not particularly druggable. We've got a few monoclonals. Is that one? Oh yeah, it needs to be resorted. We've got firatograst. It's an anti-inflammatory agent. It's got an interaction score of 2.91, which is not too bad. That could be another potential therapeutic target, knocking out ITGB1. So we've got some interesting things going in here. When the scores start dropping, so these are less good, we probably wouldn't look at those. I mean, 4 isn't bad, but we can see sometimes scores up to 20, 25, 30, and those are really strong. But often they tend to be the drug that's specifically used for that molecule as a treatment. So they're already an occupied space in terms of the IP. So we can use DGI-DB to look at that. Sorry.
Can that be used to study other type of interactions like drug-drug interaction or drug-protein interaction?
That might be better solved by Kemble, because Kemble would be drug-protein and drug-drug. This is about, well, it's protein-drug interactions because it's the protein product, gene product, which is the protein. So you could use that for gene-protein, so drug-protein interactions. But for drug-drug interactions, it's probably better to look at Kemble because that has the chemistry part of it. So, yeah. If it's drug interactions via a molecular or protein gene intermediary, then that would do it. But if it's direct drug-drug interactions chemically, then you're better off with Kemble. So that's DGI-DB. What we can also do is is we can apply these things to druggability indices as well. So DGIDB is one example, but there's also a piece of software called Drug Genome AI, or Drug Genome AI. So Drug Genome AI is from AstraZeneca, and it's a way of working out how druggable is a small molecule. So if we look at that, so it's, if we do a search on Drug Genome AI, it's served through GitHub, which is the developer approach and developer way of sharing things. Okay, how are we doing? Yep, that's good. So it's loading. Taking a bit of time. So if I click on the home, there we go. So I can search for a gene. So if I put ITGB1, oops. You can see it comes up with ITGB1. Right, so now it's interrogating this database and it has various scores. So it puts these into different models, and these are whether they have reached different phases of clinical trials. So has there been, for this target, a molecule that's been taken to a particular phase? So has it been taken to the clinical trial phase, the chemistry phase, and then you have different tiers. These tiers are defined here. So T-clin, let's see if it wakes up, is approved, clinical trial approved drugs. Yep. So my T-clin score is 97. So it's been, something has been taken to clinical trial for this molecule. T-chem, smaller molecule activities have been shown in Kemble. This approved drug targets with chemistry. Tier 1 is targets approved for biotherapeutic use, and Tier 2 genes that encode targets with known bioactive drug-like properties. So you see, as you go up the tiers, so they're more likely to be clinically approved drugs. But this is scoring pretty well with high probabilities across all those criteria. So that's making this look very suitable as a potential drug target. We also get where there are potential interesting areas of method for treating. So we've got small molecules that scores 97.8%, a raw score 99.99% for antibodies. So it's clearly antibodies, we know that, we saw those earlier. And for PROTACs. So a PROTAC is a drug modality which is drug antibody conjugates. So where you bind a drug onto an antibody, direct that at a cancer, and when the antibody hits the cancer, binds to it, it releases the drug. So it's quite encouraging for this target in terms of that. We have various scores for small molecule antibody and small molecule in the cancer and non-cancer space. So CPD clinical phase small molecule drug candidates in oncology. So we know we're targeting breast cancer, so that's a potentially interesting offering. And then this is showing the modalities where it might well work. It shows the cellular location, so it's intracellular and membrane, so that's why it's a good antibody. And there are orthologs for this in other species, so if we need to create an animal model, we know there are orthologs there, and we can see here where it's expressed. So we can estimate where other effects may occur if we start to mess with this. And this is the expression of the protein in different parts of the system. Oops. So if I don't think drug Gnome AI actually gives that. Let's have a look. Can I right click on that? No, I can't open that. But I can look at that in maybe Kemble or DGIDB, because DGIDB is doing that function primarily. Yeah. The real place you could look for that is open targets. So if you want to know the drugs that have been approved for that molecule, then you can search that in open targets. So, sorry, I'm jumping around. Yeah, so drug Gnome AI is based on Mantis machine learning. Looks at disease-specific gene druggability framework. It uses a semi-supervised learning approach based on Keras and TensorFlow, so it's a Python or R-based package ultimately. I think if you go onto the website, you can download the package for it. And then the publication is Communications Biology 2022. So it's a publication if you want to look that up. This is what I was saying about the digital twin concept. So this is visualizing it. So we have a network. That network is quantified. That network is being built for a particular data set. So we can take our P53 plus network, which we found in cancer, and then by taking those same genes into network inference in, say, healthy breast, we can build a quantified model of it in healthy breast. So now we can put the cancer pathway side by side with the healthy pathway and start to see where the differences lie. What are the differential drivers? Ideally, we want something that's a strong driver in cancer and a weak driver in healthy, or a strong receiver in cancer and a weak receiver in healthy. So that starts to help us whittle down the key targets. But what we can also do, and we've gone out, and it feels a bit like the body snatchers, but I'm doing it digitally. I've gone out and collected all sorts of healthy organ data from publicly available databases. And then I can use that to apply these network inference models in different states to build effectively a pathway model in different healthy tissues. Because basically I want to be able to drug something that's just in cancer, just in this case just in breast cancer. But I don't want it to have an effect on colorectal tissue or effect on blood or an effect on the brain. So by building these digital twin models across different tissues, I can then rank my molecules to see which ones are important in the disease, but not in all these healthy states. I'm nearly done. So we can use AI for molecular docking as well. So we can look at 3D structures, and we can look at drug molecules as structures. We can also, two ways of looking at drug efficacy. One way is to look at the structure. And if you remember yesterday, I talked about 3D convolutional neural networks being used for docking. Another thing I can do, and I've seen companies do this, is every molecule is represented by what's called a SMILES code. So that's a string of numbers, letters, which represents the unique structure of that. Now I can mine that SMILES code, if I put it in a framework, to see which elements of the SMILES code indicate something that might bind to my molecule from the literature. And then I can use that SMILES code in a model in a generative way to find new structures, new SMILES codes that might lead to molecules that could be created that haven't yet been created. So by looking at either the 3D structure or the SMILES code, I can start to build molecular docking models. Traditionally, we would get that protein structure from X-ray crystallography. So basically, we take a protein into a crystal form in a resin or a matrix. We fire X-rays at it, and based upon the shadows that are produced by the interference patterns of that protein, we can start to work out what the structure might be. Because different parts of the protein diffract X-rays in different ways. So alpha helixes diffract differently from beta pleated sheets. So by rastering across the image, across the resin bulk, changing the orientation, rastering across that, we can start to build up a 3D structure. Traditional way of doing it, but more recently, of course, we can get structures from AlphaFold and other pieces of software. AlphaFold isn't the only answer. Just because they won the Nobel Prize doesn't mean that the research in that area completely stops. In fact, there have been 5 or 6 other iterations of AlphaFold that have come out since that. Hence the word "and similar." So we could model SMILES codes with LLM and foundational AI to build some binding predictions. So that's pretty much how we can utilize this process, going from data, proteomics, transcriptomics, through to enrichment. So what are the molecules? What is the biology that associates with this question? Through to pathways. What's the biology that associates with this pathway? Through to inference. What are the most influential, most influenced molecules in this system? Through to druggability, through to drug design and drug discovery. So by doing all that workflow, we can create an in silico pipeline to find new targets at the cost of electricity, because there's that much data available out there already. But don't stop producing data, people who are using mass spectrometers, and don't stop producing transcriptomic data, because we are literally a millionth of the amount of data that really would fully represent the human system. But we can use what we've got. So in this way, the idea is we can get to new treatments for diseases much more quickly. And we can get to new drugs much more quickly. Because the conventional pipeline, as was, it would probably take us around 15 to 20 years to find a molecule that might be a drug target. And all the validation, all the knockout studies, all the studies of association through a disease model, because invariably they weren't real human tissues, they were cell line models or animal models. All of that would take around 15 to 20 years and cost a huge amount of money. Then once we've got the biological target, it would be another 15 years to probably take it to clinical trial. So that's a pipeline that takes 30 years to get to benefit for humans. The technology is moving so quickly, that pipeline could be out of date within 6 months. There's also a 95% failure rate and a multi-billion dollar cost to get a drug to market. So it's on that landscape that we're starting to do these things, because we think there is going to be a much better way of doing things using machine learning. And that's the basis for how we do things. So this is my NTU email address. I have an ARU email address, but I'll just leave that there if you want to contact me. But I'm going to be carrying on teaching you. Are there any questions? Yep. Yeah, you may have to speak up because this fan is really... To consciousness. Yep.
So basically, what Roger Penrose has told that regarding quantum gravity.
Quantum gravity, yep.
Basically, what he told that consciousness is a thing which originates from microorganisms. That can also be synthesized from the protein, but that is a debatable. Actually, consciousness is still a mystery. There are many theories. So I want to know what's your thought.
So consciousness, by its definition, is probably quite continuous. In the words of Einstein, though, how can we define consciousness when we are within that consciousness ourselves? Because we can't solve the problem of consciousness because we are within that consciousness. Although we can contemplate what a consciousness might be, but you know, if you're familiar with the principle of Tao in Chinese, as soon as we seek to define that which is Tao, then we cease to be able to define it. And could we argue consciousness is a bit like that, in that we can get so far in defining consciousness, but then by trying to define it, then we cease to be able to define it. It's a philosophical question, but I think we can replicate elements of consciousness. We can get so far towards defining it.
What I'm trying to say is like AI is getting advanced, so is it possible to solve it by quantum computer, which is already Google DeepMind is working on that?
So that's a good point. So at the minute we really have memory, but do we have consciousness? And we probably don't have consciousness because these things are incapable of self-awareness. So intelligence, or an aspect of intelligence and consciousness, is self-awareness. Is a computer, just because it's good at mapping and modeling language, aware of itself? Probably not. There has to be, and this is where it gets to be a bit of science fiction, because you know, we have this AGC, Advanced Generative Computing, AGI, sorry, Advanced Generative Intelligence. Where does the spark come in an extremely complicated computer for it to develop from memory to consciousness? That's a kind of a quantum leap. I mean, what is consciousness? And consciousness can be defined by animals, by anything at a certain level. You said about even at the protein level.
There is no measurement of consciousness.
Excuse me, sorry to interject. I think we are going a bit way more, and we have some other time. We are chasing few sessions, and today we will be finishing off our sessions at 4. So those who have registered, please proceed with the lunch. And while you're at lunch, you can discuss if you have problems, whatever you want to discuss with Professor Graham.